The second part of module number 2 deals with the isolation of airborne microbes. We discussed in the pre-laboratory the concept of air as a temporary reservoir of microbes, in which, while they are in the air, microbes are usually not metabolically active. For the most part, we can consider that air is serving as a transmission matrix, unlike soil, which is more of a residence matrix, and in a matrix where microbes are expected to interact with one another as well as with macroorganisms. So the case of air is very different. A lot of airborne pathogens are transmitted via air, usually via aerosol or droplet mechanism. And as we know, the etiologic agent of the current pandemic, COVID 19, SARS-CoV-2, is added to this growing list of airborne pathogens such as airborne viruses, Mycobacterium, tuberculosis, Streptococcus pneumoniae, Bacillus anthracis, and many more. No? So it's very important that as microbiologists, you will have a fundamental understanding of how we can sample airborne organisms or airborne microorganisms. Let's review a few things before we proceed with the discussion of the expected results for this module. So, last time we talked about how airborne microbes are considered as accidental wanderers from their original source to the air, which is a transmission area, the transmission matrix, as organisms that are carried via the air or air as a transmission matrix. We expect that airborne microbes are usually attached to carrier or vehicles. For the case of aerosols and in droplets, no? We expect that airborne microbes are should have this mechanism of being transmitted through the air. No? Aside from what I've mentioned, they may also be carried by dust particles, such as small soil clay particles. Like um, for the case of parasites or protozoan parasites that undergo encystment in the soil, diba? So they may attach to dust or soil particles, and they can be flown by air, or they can flow freely. They can float freely on the air. Some microbes can also exist as floating bodies that are surrounded by films of dried organic or inorganic materials. No? For a visual representation, let's take a look at this image. No? Viruses and bacteria here. So viruses and bacteria, they're carried via aerosols. And normally they fall between the range of ultrafine to fine. So the very small diameter, and if you would consider air as an important transmission matrix, then understanding how they can be transferred or how can they be airborne is very fundamental, especially for the cases of epidemics or outbreaks. It's very important to understand how pathogens actually develop adaptations in order to be airborne, diba? And from our discussions and from what you're hearing from the news, perhaps you have heard the words aerosols and droplets a lot. Probably you've heard those two words a lot, especially with the developments of our understanding about the biology of the COVID-19 etiologic agent. No, let's differentiate these two terms. So aerosols and droplets. I have a very nice infographic or figure from this paper comparing aerosols and droplets in the context of SARS-CoV-2 transmission. In general, aerosols are liquid or solid particles that are suspended in air. They can be visible, like fog, but most of them are often invisible, like dust or pollen. Usually, the size of aerosols will be below 5 microns in terms of diameter, and we can divide them into, in terms of their size, we can divide them into small droplets and large droplets. Large droplets usually diameter of more than 5 microns. They drop to the ground before they evaporate, causing local contamination. No? So in essence, that's how we differentiate aerosols from droplets. No? So droplets are the ones that do contact, the ones that contact a particular surface, while aerosols are purely for airborne transmission. Okay? Now, I want you to have a basic understanding of how different they are, because in our context, like if we really did the laboratory in a face-to-face setting, there will be a component in which the areas to be sampled will be put into a operational state, meaning people are are passing in and passing by within the surrounding environment where you're collecting air sample, and there will be a component where there should be no operations, or the sampling area will be free of any individual passing by. Therefore, differentiating, or giving us the idea of whether or whether operation, or the presence of an individual in a built environment for the case of the sampling sites for our activity will be affected by the frequency of individuals, the quantity of the microbes to be isolated from air. Will it be affected by the number of individuals within the sampling site? So it's important that we distinguish the difference between the two, between aerosols and droplets. No? Okay. For the case of COVID-19, for the case of SARS-CoV-2, okay, we can appreciate that, in we can appreciate that for the case of airborne transmission, aerosols are the primary method, the primary route. So, therefore, the need for protective coverings in order to prevent the transmission of such aerosols from the donor to the recipient. Diba? For the case of droplets or contact transmission, the probability or the likelihood of infection from a donor will be less compared to airborne transmission. So it's the problem is actually more with airborne transmission, especially during the summer, because it will allow for aerosols to have enough or have an optimal environment where they can just freely float in the air and travel far distances from the donor to the recipient. On the other hand, like in the case of wet season, like draining season, droplets are problematic compared to aerosols. So, discussing that, I have attached here, or mentioning that that narrative, I have presented here the mitigation paradigm presented in this paper. So, several scenarios of virus transmission under the different mitigation techniques, or under mitigation limits. No? So, we have for path A, if social distancing is implemented, how strong is airborne transmission and contact transmission. You see that contact transmission will become lesser, while airborne transmission doesn't really stay or doesn't really decrease. No, that even though given social distancing, airborne transmission of aerosols is still of the same magnitude. On the other hand, for path B, if we add social distancing and face covering, as well as what they call it, as well as quarantine and isolation. Okay, so uh, there will be a difference in terms of the transmit in terms of the likelihood of transmission. There's decreased transmission in terms of contact tracing still from social distancing, and further decrease by face covering. If there's quarantine and isolation, there is no transfer at all from donor to recipient. No? Given that there is also testing and contact tracing. Then lastly, for another varied scenario, we have face covering first before social distancing and then quarantine and isolation. You see how effective the combination is. No, so both aerosol transmission and droplet transmission is decreased by a very significant portion if people wear masks in order to prevent any aerosol from any aerosol from being released or droplet from being released to any contact surface. And provided their social distancing, the more decrease in terms of the likelihood of transmissibility, and quarantine and isolation definitely will prevent transmission from donor to recipient. So you see how important the mitigation limits are, from social distancing to protective covering, personal protective equipment for the case of the airborne SARS-CoV-2. We have our face masks, and then quarantine and isolation as um, uh, a very effective way of ensuring that donors or no more recipients will get infected. No. Okay. Here, I ask you to read this review paper on the drivers of airborne human-to-human pathogen transmission. I hope that you were able to skim through the article. It's a very nice work. So you will be able to gain a fundamental understanding of how airborne transmission is a very important trait for pathogen dissemination, for pathogen distribution in our environment. So this paradigm or this framework shows us the circle of events leading to human-to-human transmission of airborne pathogens. So initially, number one, the pathogen is, of course, associated with either small aerosols or droplets or dust particles when they are transported from the donor to a recipient. So there's association with the carrier, the vehicle. Could be an aerosol or could be a droplet. No? This may be directly or via an intermediate stage, normally involving settlement on the surface and re-emergence into the air later. Like for the case of droplets, the idea of settlement is very important because in cases where if you do disinfection of of a contact surface, then whatever settled on that contact surface will definitely what do you call this will definitely die if you do disinfection, diba? Any settled droplet will die and will have no opportunity to re-emerge in the air. Okay, then a low infectious dose, we expect that because of course there's an intermediary, especially for the case of large droplets. Low infectious dose will be sufficient enough for deposition of the pathogen from the donor to the recipient. After infection of susceptible cells, pathogens may amplify at the site of the deposition, what we call the primary site, no? And or they may also affect secondary or peripheral tissues. No, they may disseminate from the primary site to a secondary site. No? And eventually, when the organism, the pathogen, is able to multiply, it's now not welcome anymore in the body of the recipient because of let's say the recipient was able to develop the immunity against it, and the immune system is capable of detecting the pathogen already and expelling it out of the recipient's body. That's a time that the pathogen will try to or will begin its search for an exit portal. So it will exit from the body of the recipient, and essentially the recipient becomes a donor, no? And the process begins, no? Or the cycle actually repeats, provided that there's an interruption, like for the case of, yeah, like what we've mentioned in this different scenarios. If there is interventions along the way, then we can prevent this cycle of infection and or reinfection, no? So for module number 2, what we did are two things, or what we implemented are two techniques to isolate airborne microbes, no? From airborne microbes from because from air, no? So we employed settle or gravity plate method, and we also employed the impingement method. Okay, so we're going to discuss both of them, both of these techniques, as we proceed with our discussion now. Okay, so passive monitoring using settle plate or the gravity plate or the sedimentation technique. No? Passive monitoring uses settle plates, that you can see here, which are standard Petri dishes containing culture media. And the idea is that we expose them to the air for a given period of time in order to collect biological particles, which we call the sediment, no? And afterwards, we incubate these Petri plates. The results are usually expressed in colony forming units per plate per time, or colony forming units per area per hour. So two factors are considered, no? So we can consider the settled plate itself as a quantitation module, as a quantitation element, as well as, and then we factor in time, or we can consider the area of the location, the area of the sampling site, and then the time, no? So according to a few authors, passive sampling through the settle method or the gravity plate method is a valid risk assessment measure, no? Because it provides us the information regarding the viable airborne pathogens that are landing on a critical surface. Like for example, you want to monitor the microbial load of a surgery room, okay, an operation room. So a surgery operation room, of course, there's the operating table, and if we want to know like what microbes are usually landing on the operating table, we can apply the passive method, the settle or gravity plate method. No, another is in theaters. We can also do monitoring of the microbial load of air in theaters using the gravity plate method. Okay. As I mentioned nga, we can quantify microbes sampled from the air using the gravity plate method. We can express them as CFU per plate per time or CFU per area per time. Now, the validity is some people or some microbiologists may contest the validity of settled plate. But probably it will really depend on how the method is executed or how the technique is executed. But given everything considered as given everything controlled, no, the method is valid enough in order to tell us whether viable organisms that can cause disease land on a particular surface where you put your settle plates. No? Especially essential surfaces that should not really have such pathogens. No? I mentioned about the applications already. So there are a number of applications, and in comparison with what do you call that, with impingement, no? So settle plate is less expensive, doesn't require secondary materials, and it's really time saving, no? Compared to um, the impingement method. So in the paper, in the worksheet, I ask you to decide on your own the number of colony counts per type of culture media, and then you provide me a certain discussion that will justify your observation. No, so in essence, there are a lot of factors to consider. So this one will be very high. The answer to this will be very individual, and I will just I will consider your explanations if you wrote them well. If you considered all the fact or majority of the factors that will affect the distribution, the number, quantity of these colonies per culture media per location. No? So we have two extramural rooftop and street, and then two intramural locations, no, where you intended to sample airborne microbes. No? Now, what can we expect to see? No? Let's just take a look at some of the microbes that we expect to see. Because I also asked you, one part of the mode of the worksheet is for you to draw or provide pictures of two bacterial species and two fungal species that you expect to see in these spaces, in these extramural and intramural spaces after air sampling. Let's take a look at some. So one is Bacillus anthracis. Bacillus anthracis is the etiologic agent of anthrax, a common disease in livestock and occasionally to us humans. They're obligate pathogens within the genus Bacillus, and the disease that they carry is classified as a zoonotic disease, causing infected animals to actually transmit the disease to us. So if there's an infected cow with Bacillus anthracis, spores coming from them may, travel through air, and we can inhale those spores. If our immune system is not strong enough, that we may develop anthrax. No? As an airborne infection, primary route of Bacillus anthracis is via the inhalation of anthrax spores. No? If you grow them in blood agar, they will look like this, no? Bacillus anthracis. And if you look at them under the microscope, they will look like this. Okay, gram negative. Then another example is for the case of fungi, Aspergillus species, which is a very diverse genus of molds. No, so there are a few examples here that I have given. When you look at them at the colony plate or the PDA, potato dextrose agar plate, normally they will look like this. Oh, they have certain pigmentation. The mycelium may cover a portion of the plate or the whole plate depending on how long you've incubated your sample. If you do your smearing using lactophenol cotton blue or any other dye, then you will be able to appreciate the hyphae and the reproductive organ of the organism. You can see it. And as I mentioned a while ago, yeast cell or mold cells are looking like plant cells now when you observe them under the microscope. So I just hope that in your worksheet you were able to provide me with expected fungi and bacterial species to be isolated from soil. Okay. Now we have active monitoring using the impingement method. Okay. The liquid impingers are one of the most practical techniques for sampling bacteria, particles, and fungal spores because we can sample large amounts of air in relatively short periods of time. If you've seen the video, then you by now you know that using a liquid impinger will give you the benefit or the advantage of sampling a lot of air in terms of volume, no? For a whole area. So this is we call it active monitoring because we have the introduction of pressure, right? So our equipment sucks in air and impinges the whatever component is sucked with air into the liquid, so that from the liquid we can grow, we can culture subculture whatever is impinged or whatever is mixed in the liquid sample, in the liquid impinged material. No, okay. If we are going to compare the two methods, as I mentioned, settled plate is less costly, while for the impingement, a lot of things. There are also benefits or advantages that you cannot really get if you use the settled plate method. No? But the principle is very clear. Unlike sedimentation, air is drawn through a small jet and directed against a liquid surface so that the liquid will sort of extract the particles from the air. No? It's very suitable for measuring viable organisms and the concentration over time. So we have the factor of time in impingement liquids or the impingement method, and it's more I'd say a lot more commonly used, unlike the settle plate method. The rate of collection will actually be varying. It will depend on the orifice of our this one. It will depend on the orifice of our tube. So whatever is like the rate of uh, air suction, no, okay, will actually be dependent. That will actually dictate the rate of collecting the sample. There's auxiliary or secondary equipment needed a lot actually, so it's not really time saving, and it can be costly compared to sedimentation, compared to settled plates. Okay. So in our module, you are tasked to quantify as well, diba? Similar with microbial sampling from soil, no, colony counts are asked to be computed for the standard plate counts for both method. I mean for the ano lang pala, for the impingement method only. No? The number of liters per sample, cubic feet of air sample, and colony forming units per cubic foot of air. Those are the three things that I hope by now we have understood how to compute. So the number of liters per sample, as I mentioned, is dependent on the sampling time, how long did you do your sampling, and the capacity of the limiting orifice, normally expressed at this particular value or this particular unit, liters per minute, or it could be volume per time, no? So a specific rate for the limiting orifice. So you just have to multiply your sampling time with the capacity of the limiting orifice so that you can get the number of liters per sample. No? Now that number of liters per sample, you have to convert that to cubic feet of air. Normally, 280 liters of air will be equivalent to about 10 cubic feet of air. So we were using the factor here 28. So whatever you get for from number A will be divided into 28, and that's your cubic feet of air sample. Okay, and that will be your value for VA. Okay, so your number of your standard plate count will then be equivalent to the number of colonies per milliliter of the impingement fluid multiplied by the volume of the impingement fluid used. Since we did spread plating, we do 0.1 ml. If you did pore plating, then you do 1 ml. Then you just divide it with the cubic feet of air that you sampled. The table shows us for bacterial counts and fungal counts. So I have two tables here. And similar with what we've already discussed for soil sampling, the same expectations or same expected results should appear. Like for 10 to the negative 2, colony count should be lower than 10 to the negative 1. And for fungal counts, the same applies. Okay? 10 to the negative 1 should have greater 10 to the negative 2 counts. For the standard plate count, as I mentioned, I have already prepared an automatic calculator so I can check your answers later on. Okay, the guide questions, most of these questions I have already answered all throughout the presentation. No? So in general, which microbial groups is or are expected to dominate in each soil type? You just have to justify your answer. Basically, actinomycetes are the major dominating bacterial group in soils. And for different types of soil, it's kind of hard to answer that because we're only given with a limited description of what the soil is. Unlike if we really did this in the laboratory, then we can appreciate more in terms of empirical data. But in essence, I've already mentioned that that microbes will tend to have lesser distribution if the pH is extreme. No, so if your pH is very acidic or very basic, you don't expect microbes to survive that well. So most soil microbes will really thrive in pH between 5 or 6 and 7. 5, 6, and 7 are good values for the pH. And we know that through time, soil acidifies. The pH of the soil acidifies, not only because of time, but a lot of factors, especially breakdown or decomposition of organic matter. Now, second question, how do pH and moisture content affect the predominant microbial types in the different soil types? I've already answered that. For the case of pH. For the case of water, so we just have to I must be able to see from your discussion or from your answer the importance of water as a matrix for reaction. Diba? So water serves as a solvent for chemical reactions. And we know that microbial communities, microbial cells, even though very small, are expected to undergo very numerous interactions and chemical reactions. So water is very important in order to facilitate such reactions. No? That in the absence of water, only those that are adapted to dry, arid conditions will survive. Third question, what's the function of rose bengal in potato dextrose agar? So by now, you know that PDA, potato dextrose agar, is intended for fungal isolation. Now, rose bengal is actually a dye, and we use it to inhibit the growth of bacteria. So if you really want to grow, if you just want to grow or isolate fungi from your soil samples, you have to use rose bengal to inhibit the growth of bacteria. And then in some types of culture media that contains rose bengal itself, it serves as a what do you call this, as an enhancement for the growth of the fungi itself. So rose bengal is an enhancer as well for the case of fungi isolation. Okay, and question number 4, differentiate actinomycetes from other members of the domain Eubacteria, and why are they abundant in soil? So this question, I've answered the first part. So we've discussed about the morphology and characteristics of actinomycetes in relation to actinobacteria. So we know that actinomycetes appear, though most of them will, or we expect that they appear as gram-negative. Their cellular arrangement is quite different. No, um, they form strings or filaments. Uh, as we observe, if we observe them under the microscope and we do gram staining, we expect that they have what do you call this? They have filamentous, stringy appearance. No, okay. In terms of colony, like when you observe it under the colony or under the plate itself, the colony appearance is different. No? So it's leathery, powdery, much similar to fungi, though not really fungi in terms of the totality. The colonies of actinomycetes will normally produce very dry colonies, very brittle colonies, no? That may or may not impinge through the soil agar or the agar medium. And then the second question for question number 4, follow up. Why are they abundant in soil? Primarily because actinomycetes are very flexible. So they are metabolically flexible. Some of them are aerobic, some of them are anaerobic. So they can, what do you call this? They can what do you call that? They can grow in the different pockets of the soil, even though there's no uniform pattern or no uniform distribution of characteristics or parameters. Because of the metabolic diversity of actinomycetes, we expect them to be abundant in the soil. Also, they're metabolically competitive. As metabolically competitive group, actinomycetes will find the soil a very nice environment to biodiversify, because not only they will encounter other microbial species and communities, but also they will encounter macroorganisms such as plants and animals that they may develop certain associations or relationships with. Okay? Fifth question. When is the most probable number method preferable to be used over the standard plate method? Now, let's define what MPN or what the MPN method is. The MPN method is actually a statistical technique used to estimate viable numbers of bacteria or microbes in a sample, normally by inoculating broths in tenfold dilutions. And it's actually based on the idea of extinction of dilution. No? So, if you've had your general microbiology classes, I'm sure that you did your laboratory already, you will remember that MPN is applied to check for the quality of water, diba? So MPN is applicable if you want to check viable cells in water, in liquid form, or in food. No, unlike for the standard plate count, in standard plate count, mostly we're dealing with solid media, or like for the case of air that's also employed, no, standard plate. So more solid media or more solid sample compared to most probable number, in which we expect that the viable cells are suspended in a liquid matrix or in a liquid culture media. Then six, what are the advantages and disadvantages of employing settle or gravity plate versus impingement methods in estimating the number of microorganisms in air? I've discussed this already. Some might consider the gravity plate to be less valid compared to impingement. Well, both of them are valid methods. Now the execution will really be the deciding factor of whether the results of a gravity plate employment or an impingement method employment will be different or will be significantly the same. No. In terms of advantage, we expect that both of them have differences. So for the case of settle plate, diba, where we talked about critical surfaces. So they're very applicable if we want to assess how critical surfaces are populated by airborne microbes. On the other hand, we cannot do that with impingement. In impingement, we are doing active monitoring. So for impingement, one advantage is we can sample a lot of air in one sitting. So we can really sample every air in the environment, diba? Given that it's actively including a pressure pump in order to suction in air and then filter or extract particles from it using our impingement fluid. No? Last question, do intramural environments always carry less air microbial load than extramural ones? Explain some factors. Not all the time, no? The answer to this is very obvious. It's always context dependent. Even though intramural environments are closed environments, we don't expect them to be we don't expect them to be free of these microbes that are suspended in the air. No? So we expect that in both areas or both types of environments, the distribution, the number of airborne microbes might be similar or different depending on certain conditions. Like for the case of operations, no? If a certain intramural environment is being used, then you expect that the airborne microbes can be greater in number compared to the moment that it is not used. No. So that's just one factor, operations. A lot more, I expect to read more of the other factors affecting the abundance of microorganisms in the air on your worksheet answers. No. But again, as an answer, this is very context dependent, the answer to this question. Okay, so that ends our session. All the references I've used are enlisted here. You can see the syllabus for a complete list of the references as well as the articles, then I have some insets per slide that I hope that you will take note if you want to read more about those papers. Okay, so that ends our session for the post-lab discussion for module number 2.